Yes.
Okay. Okay, so thank you very much and good afternoon. And first of all, I would like to thank Gurbooth for this invitation in this Work Group 5. And the main aim of this presentation is to demonstrate the importance of considering availability of the nutrients when we are optimizing insect feed, as well as to present a method that we have developed for determining digestibility in yellow mealworms. Okay, the next one. I'm currently responsible of this AnyFed group, as has mentioned Gurbooth, at the Institute of Animal Science and Technology at the UPB. And the main aim of this group is to study and optimize the animal nutrition, which constitute the main cost of for livestock farms, including those in the insect industry. Our team research focus in three main areas. The first two are not related with our activity. The first one is developing tailored feeding strategy using precision livestock farming tools. The second one is generating solution for endangered wild species using animal nutrition tools. But the third one is to develop sustainable protein alternatives, and we are working on insect nutrition, but also in the use of solid state fermentation to enrich byproducts, and also in the generation of synthetic proteins to be used in animal nutrition. Okay, respect to this presentation, the first question this presentation will address is that why it is important to determine the stability of nutrients in insect. This technique is widely used for all livestock species to determine nutrient availability, but is it really necessary, for insects such as mealworms? Okay, we are all completely agreed that at least larvae require energy to support maintenance and growth, and we understand this because we all provide a diet, not to be transformed into larvae and frass over several weeks. But we know that larvae need energy, which usually comes from digestible carbohydrates in form of sugars, such as glucose, for example. And these sugars are transferred or transport to the cells where are oxidated to generate ATP, our cellular energy currency. But also energy comes from fats, which can be converted into acetyl coenzyme A through beta oxidation, and this is all converted in ATP in the Krebs cycle. Okay. In the case of the protein, larvae also need proteins to build their structure and generate the enzymes and metabolites that carry out most of the cellular and physiological functions. However, they really need the 20 essential amino acids that make up these proteins. No? We also need fiber, something very important, to maintain a symbiotic relationship with microbiota, which improves dietary utilization and promotes larval health. And also, in the case of mealworms, these fibers also often serve as a bedding as a litter for mealworms, enable larvae to move and to grow properly. To optimize the growth and health and well-being of our larvae, we also need to provide them minerals, trace elements, vitamins, and other nutrients such as nucleotides, some essential fatty acids, steroids, etc. Okay. However, the definition of nutrient requirements is often incomplete and inconsistent, resulting in a wide range of results in the literature. A common way to define the nutrient content in our diets, for example, is carbohydrate to protein ratio. Okay, but what exactly do we mean by carbohydrate? There are many types of carbohydrates. What about the fats? Should they be left out of the equation? We have also considered, for example, the carbohydrates and the proteins provided by the moisture sources, such as carrots, or only are provided by those provided by the dry food. Sometimes we don't use the same nomenclature. Okay, is everything of the larvae that the larvae consume finally converted into available nutrients, or is some lost? Okay, or for example, some question is, are we taking into account the contribution of our microbiota, of the gut microbiota of the larvae? Some nutrients are transferred and fermented, and the availability of the nutrients changes as a consequence of microbiota. Have we considered this effect? Okay, so all these type of things appears, and for example, in the case of carbohydrates, these carbohydrates can be classified mainly in two types: soluble carbohydrates, sugars and starches mainly, which are really available energy source for living beings, including insects. And the other type of carbohydrate is fiber, which consists the structural carbohydrates mainly from the cell wall of the plant, and is generally much less available as an energy source. However, fiber is very, very, very complex. Within the total dietary fiber, the total fiber, there are more fermentable fractions such as soluble fiber, fructans, mannans, pectins, and others, and less available fractions such as neutral detergent fiber, well known as NDF, and even within this NDF, there are more and less available fractions. For example, more available ones include hemicelluloses, insoluble hemicelluloses, and less available ones include cellulose and lignin, being recommended a complete analysis of this type of fibers using the advanced methodology. This is particularly important in the case of our insects, since we usually offer our larvae a wide range of byproducts with a fairly high total dietary fiber content and a very diverse fibrous nature. For example, in this slide illustrates the importance of this issue. In the figure on the left, shows the carbohydrate content of three typical mealworm raw materials: barley grain, hominifeed, and wheat bran. Okay, the total carbohydrate content of these feeds is quite similar, no? The total height of these bars, 66, 67, and 62%. However, in the figure on the right, it shows the digestible energy content for these same three raw materials that we have determined in one of our papers. And it reveals that barley grain contains almost 60% more available energy than wheat bran. This is mainly due to the higher amount of soluble carbohydrates in the barley grain, in the yellow bars, compared to the wheat bran. However, even with a similar soluble carbohydrate content, okay, even hominifeed, which contain more soluble carbohydrates than the barley grain, okay, has almost 20% less digestible energy, since the starch of this hominifeed partly retrogrades as a result of its industrial treatment. Okay, this highlights the need to measure available energy in ways others than by carbohydrate content alone. Okay. In the case of fiber, the effect of fiber on the digestible energy content of the diet depends also on its nature. In the figure on the left shows the NDF levels of various fibrous raw materials, as you can see, which range from 36 to 49%. We can also see that the higher is the hemicellulose content, the most soluble fraction of the NDF, represented in the yellow bars. The higher is the digestible energy content of this raw material in question, as you can see in the figure on the right. Okay, so fiber nature is very, very important. And that's not enough. We must also take into account the possible effect of microbiota. In this figure, and especially in the formula that I provide, we can see that the digestibility of the ADF, acid detergent fiber, the less digestible fraction of the fiber, depends primarily on its own content in the diet, and the content of the ADF. You can see here, okay, sorry, here. Okay, the stability acid depends on the content of the ADF. The more is the ADF in the diet, the more digestible it is, since the higher cellulose content likely favors the development of cellulitic microbiota and therefore facilitate the utilization of this cellulose. All of this without considering the contribution of the fats. Okay, that's why defining the energy contribution of the diet cannot be based on the content of the specific nutrients, such as carbohydrate, starch, or fiber. We must focus on digestible energy because calories are standardized. It comes from independently of the origin of these calories. We standardize the actual amount of energy available to the insect. The same thing happens with the protein. In the case of the protein, in the figure on the left shows the crude protein in yellow and the digestible protein content in orange. Of three common sources of protein: wheat bran, corn gluten feed, and soybean meal. As you can see, the digestible protein content and therefore the availability of the available protein supply depends mainly on two factors. The first one is the crude protein content. Generally, the higher protein content, the higher the digestible protein content. This is evident, for example, in the case of the soybean meal, which has a high crude protein content, 52% in yellow, as a dry matter basis, and a correspondingly high digestible protein content, 24% in dry matter basis. However, it also depends on the digestibility. For instance, although the wheat bran contains less crude protein content than the case of the corn gluten feed, the digestibility of the corn gluten feed protein is very low in comparison to the wheat bran. Finally, both raw materials provide the same amount of digestible protein, this 7% in dry matter basis. Thus, the availability of the protein provided is also important to take into account. In conclusion for this initial presentation, insects require readily available nutrients. Therefore, when discussing the characteristics of the different raw materials, it is recommended that we do show in terms of digestible energy or digestible protein. Essential amino acids available or fatty acids, available digestible essential amino acids or fatty acids, as well available minerals, trace elements. These available minerals already being done in some species like broilers, and also vitamins. For this reason, we should have a procedure to determine nutrient availability, nutrient digestibility. Okay, to determine the nutritional value of a diet using digestible energy as an example, we calculate the digestible energy content of the diet as the sum of the digestible energy content of each ingredient multiplied by its inclusion level, as in this equation. To determine the digestible energy content of an ingredient, its raw energy content must be firstly determined and then multiplied by its digestibility coefficient for gross energy. And finally, the apparent energy digestibility coefficient of the energy of this for this ingredient is determined as the gross energy ingested minus the gross energy excreted, all divided by the gross energy ingested. And if we want to express this as a percentage, must be multiplied everything by 100. In our method, we use a climatic chamber with a constant temperature of 26 degrees Celsius and a relative humidity of 60%. And to ensure consistent results, it is widely recommended that the different repetition of each diet are distributed evenly throughout the chamber, as height in the chamber and the proximity to the door or to the equipments can affect the outcomes that you obtain. To obtain consistent data, we recommend to carry out multiple replicates. This is very important. Replication is key. Based on the result of our previous trials, we recommend a minimum of 5 to 6 replicates to detect as significant those relevant differences that usually are 5% between the different treatments. To ensure a representative population fills the entire space and accesses all the food, we also recommend to use trays with an area of approximately 200 to 250 square centimeters and an initial density of 0.23 grams of larvae per square centimeter. In our case, we have used mealworms of 46 milligrams, which are large enough, but no pupating during the trial. This is important because if the pupae appears, you have a problem, okay? And the raw material to be evaluated is placed in the tray and also 20 grams of agar gel per day and tray, containing 1% of micronutrients, a mix from chickens, as a hydration source. The trial must be maintaining till you obtain sufficient quantity of RAS in order to ensure a representative sample and also to have enough to all the analysis that you will need later, okay? In our case, it lasted 6 weeks, but it will depend on the size of your trial, the amount of larvae, the amount of feed you will provide, okay? Involving weekly feeding and fecal collection. It is very, very important to provide a small amount of the raw material to be tested. What means enough to feed the animals, but not too much. So that approximately when you arrive to the 6th, 7th day, all the food has disappeared, no food remains, and only feces are present to ensure that you are only collecting the feces at the end, in the 7th day. Related to the trays to be controlled, I recommend to control the initial and the final weight of the larvae in order to ensure that the trial goes in the right weight with a good growth. Of course, the total raw material provided and the total feces collected in order to determine the digestibility of dry matter. And then we need to analyze both the raw materials and the feces for those nutrients that we are interested in. In our case, dry matter, acids, crude protein, ether extract, gross energy, and the fibers is not common, but if you are interested to know the behavior of the use of the fibers could be also interesting. In one of our recent studies, for example, we determined the nutritional value of 6 raw materials of interest in mealworms: barley meal, corn meal, hominy feed, wheat bran, corn gluten feed, and soybean meal. As you can see, the first 3 are characterized by a high amount of starch, the wheat bran by a high ADF content, and finally the corn gluten feed and soybean by a high protein content. As you can see, here appears the digestibility coefficients that we have obtained for all these ingredients for dry matter, energy, ether extract, and protein. As you can see, something interesting, the digestibility of dry matter and energy is quite similar. That this is something that could be easy for us to determine and to estimate the digestibility of energy in the future. And also that while cereals, for example, the stability of gross energy is close to 75%, in the case of other sources like gluten feed, the stability doesn't reach to 40%. Something similar occurs with the protein digestibility, which vary between 28 to 60%, depending on the source that we use. So there is a great availability of the nutrients availability in function of the raw material that we are using. With this digestibility coefficients, take into account these digestibility coefficients and also the gross energy and the crude protein of each raw material, we can determine, as I have explained it in the equation, something that we commonly call as nutritional value, determined by the digestible energy and the digestible protein, the 2 main pillars of the nutrition of all the animals. And you can see, therefore, that cereal meals is a great energy source, but they have low protein by high digestible. The mealworms has a great ability to obtain profit of the protein of the cereal, but the content is very low. The soybean meal has a great protein source, but the medium digestible. We need perhaps to find additional sources of protein. Whey bran has a low energy and low protein, but has a medium digestibility. The mini feed provides less energy and protein to that expected medium digestible, perhaps related to the treatment of this when were obtaining this byproduct. And corn gluten feed, something very frequently used in broilers, for example, has a very, very low nutritional value in the case of the mealworm. So we must reject this type of raw material. With these values of digestible energy and digestible protein, for the first time, we can start to formulate diets knowing the digestible energy and the digestible protein of this diet. And then we can use this diet in order to determine the nutritional requirements of this animal. In fact, we have recently submitted to GIF a paper to be evaluated where this requirement has been determined in a multi-phase procedure. And as a conclusion, because I believe in 21 minutes, more or less, raw materials with similar nutrients levels lead to different performance. So those things like carbohydrate to protein, something like that must be avoided. It's not very useful to talk about nutrients. It's better to use available nutrients. And for this, we need digestibility procedures. This will propose a digestible method that is not perfect. I know that it's not perfect. But if we propose a method, this one or other, that we all accept and use, we will have comparable nutritional values that we can share without problems. And using this type of methodology will be the first time in defining the nutritional requirements. And if we define the nutritional requirements, we can optimize the feeding of this type of mealworms. Okay. And that's all. Thank you very much for your attention.